Good afternoon, grade 11s. So carrying on with the urinary system, and we're on lesson 3. So I hope that you learnt the work on the nephron and that you understand the nephron, because if you didn't, then you're not going to understand the next little part. So I'm going to go back and just quickly revise the work that you did last lesson on the nephron. So remember that the nephron is the functional unit of the kidney. In other words, it's the part of the kidney that does the work of the kidney. And it is made, each kidney is made up of many hundreds of thousands of nephrons. They're very tiny, they're microscopic, and they've got lots of different regions that perform different functions. So let's have a look and quickly go through as a revision. Okay, so remember that each nephron is actually a filtering tube, and it consists of 2 parts, and those 2 parts are a Malpighian body or a renal corpuscle and the renal tubule. Now, the renal corpuscle, remember, is made up of 2 parts, the glomerulus, which is the little knot of capillaries inside a hollow double-walled cup named Bowman's capsule. So Bowman's capsule is the filter funnel. Don't call it that in a test or exam. And it's got 2 walls to it, and there's a space between those 2 walls. So Bowman's capsule plus glomerulus equals a renal corpuscle. And then the tubule consists of a number of different regions. The proximal convoluted tubule in the cortex, the loop of Henle with the descending limb and the ascending limb of the loop of Henle, and they lie within the medulla, within the pyramid of the medulla. And then this mouse keeps on disappearing, it's driving me insane. And then the distal convoluted tubule, and remember that that lies within the cortex. And the distal convoluted tubule joins with other distal convoluted tubules and forms what is known as a collecting duct. Remember that the end part of the collecting duct is called the ducta Bellini, but you don't have to know that. And the collecting duct gets bigger and bigger and bigger as it goes deeper and deeper into the renal medulla, and then the end of each collecting duct goes into a calyx. So remember that what happens is blood is filtered in the Malpighian body. The filtrate goes along the renal tubule with changes happening to it as it goes along the renal tubule, and then finally urine passes into the collecting duct. Okay. Right, so let's go, we did that. Okay, I just want to show you a little thing that you must be aware of and careful of if you get a drawing of a nephron. Now, so far, I have shown you a nephron as being, here is the Malpighian body. It leads into the proximal convoluted tubule, which loops, and there's the distal convoluted tubule. And so this is sort of a hairpin-shaped structure. And it's very easy for you to see the different regions of this. But in reality, in the kidney, it might twist over. And so I'm just warning you. It's still exactly the same thing. And I chose this particular drawing because it's really nicely colored for you to see the various parts of it. Okay, so let's have a look at this here. So this then is a nephron that has done the twisting over part that I'm warning you of. So if you look here, there's the proximal convoluted tubule. Okay. And you see how it goes behind, and the distal convoluted tubule is over it, like that. So it's almost almost as if you've taken that little U-shaped hairpin structure and twisted it over. So be careful, because if they ask you a question which requires you to know the various parts and the functions of the various parts, but you've identified them incorrectly because the drawing gave you a twisted over one, and I must admit, I'm feeling quite sorry for you guys doing online learning. I don't think I would give you something as mean as that, but just in case I forget by the end of the year. Okay, so what you need to do is, and if you don't understand what I'm saying to you here, you need to question it. Okay, so start with the thing you know. Start with the renal corpuscle and follow it along. And that first part is the proximal convoluted tubule. Whether it's flipped over or not, that's the proximal convoluted tubule. There's the loop of Henle. There's the distal convoluted tubule. And there's the collecting duct. Okay, I'm just letting you know, just in case. And I know that many of you go and do a lot of extra reading, ha ha ha, just in case. Okay, all right. So this then shows you two nephrons, and what you can see is the line where the cortex actually is. Okay, this particular nephron is further up within the cortex than that particular nephron is, and some of them are and some of them aren't. Don't obsess about it, just learn the rule. Proximal convoluted tubule, distal convoluted tubule. Remember, Piggian corpuscle is in the cortex and the loop of Henle, and the collecting duct is in the medulla. And you can see nicely here that this collecting duct actually leads directly into the center of the calyx. Okay, all right, so I'm going to show you again. So this then is a nephron in position, and here it is showing you what appears to be an upside down nephron, but it's not, it's just in a different part of the kidney. So there's the Malpighian corpuscle, there's a proximal convoluted tubule, descending limb of the loop of Henle, ascending limb of the loop of Henle. Did I call that the distal? I think I might have. I mean proximal, sorry, I'm trying not to sneeze. Distal convoluted tubule, collecting duct. And there we have collecting duct leading into the calyx. Okay. All right. So now we're going to go back and we're going to discuss the actual structure of each of these little parts in more detail. So remember, I said to you that I was going to do it simply, then I was going to do it a little bit more detailed, then I was going to do it even more detailed. So here we go. Okay, so the Malpighian corpuscle, or the renal corpuscle, lies as a little dot within the cortex. If you use quite a high magnification hand lens, you can actually see it. And I said to you that it consists of a double-walled cup formed where the end of the tubule is invaginated. So if you picture taking a glove, and the finger of a glove is like the tubule. It's closed at the end. And you, from the outside, push your finger down into the top. Does this make any sense? Into the top of a closed finger of the glove. Then it would form a little Bowman's capsule. I hope that made sense. It made sense to me, but I could picture it as I was saying it. So the cup, the double-walled cup, is Bowman's capsule, and the capillary network in Bowman's capsule is the glomerulus. Okay. A small arteriole, in other words, a branch of a branch of a branch of the renal artery. So it's an arteriole. It's called the afferent arteriole because it's bringing blood into, and that's what afferent means, going in towards something. Brings blood into the glomerulus, and it divides into many little capillaries, and that capillary network is the glomerulus. The capillaries of the glomerulus join and form the efferent arteriole leading out of the glomerulus. Efferent means leaving, going away. Now, this is critical. The bore, and that means the diameter of the lumen. Now, we've discussed lumen before. So the diameter of the lumen, correct scientific term for that is bore. And the bore of the afferent arteriole is greater than the bore of the efferent arteriole. That means more blood goes into the glomerulus than can leave. That results in a very high pressure within the glomerulus. And that pushes the small molecules, I'll talk about them again later on, through the walls of the capillaries into the space of Bowman's capsule. So that pressure is important. You must remember, bore of the afferent arteriole is bigger than the bore of the efferent arteriole, ensuring that more blood can go into the glomerulus than can leave. That sets up a pressure in the glomerulus. Okay. The walls of the capillaries are made of a single layer of squamous epithelium, as capillary walls are. And those cells of squamous epithelium or squamous epithelium, I don't care how you pronounce it, obviously rest on a basement membrane, because you learned in grade 10 that all epithelial tissues rest on a basement membrane. There are tiny pores between the cells and also going through the cells of the capillary wall, and because they're tiny pores, they're called micropores. I hope that makes sense. So they're pores, micropores, very tiny pores in the walls of the capillaries. Okay. All right. In addition, this inner wall of Bowman's capsule is also made of squamous epithelium. In other words, a single layer of very flattened cells. The cells of this epithelium also rest on a basement membrane, because by definition, that's what epithelial cells do. But these cells are very specialized. They're modified to form cells called podocytes. Podocytes are very specialized cells. They're not as simple as normal squamous epithelium. Your textbook has quite a lot of detail on them. You don't need to know any of that detail, so don't go and learn it. All that you need to know is that podocyte cells are irregularly shaped. They look a bit like a starfish. And where one podocyte and another podocyte lie adjacent to each other, their little arms sort of extend into the gaps between the other, so they fit into each other. But they're tiny slits between the cells in the podocyte layer. And that's important. Okay, so going back to the structure of the capillaries and the structure of the Bowman's capsule inner layer. So they're micropores in the squamous epithelium of the capillaries. Those squamous epithelial cells rest on a basement membrane. There are slits between the podocytes of Bowman's capsule lining. Those podocyte cells also rest on a basement membrane. So what you've got is blood in the capillaries. Blood in the capillaries. Then you've got a layer of squamous epithelial cells that have got micropores between them and through them. Then the layer of basement membrane that belongs to the capillaries. Then you've got a layer of basement membrane that belongs to Bowman's capsule. Then you've got the podocyte cells. And between the podocyte cells, there are little slits. I hope that makes sense. Okay. Email me if it doesn't. There is a drawing somewhere else. I'm 100% certain I put a drawing in here. Okay. Let's talk about blood supply of the nephrons. Okay, so I've spoken to you about the fact that the renal artery divides and divides and divides into many tiny arterioles, such that each branch of the renal artery gives rise to an arteriole. You can see branches coming off of this as well. And one afferent arteriole, which is incredibly tiny, leads into each glomerulus. And the glomerulus is a network of capillaries made of branches of the afferent arteriole. They all join together and form the efferent arteriole, which leaves the glomerulus. Now, I haven't spoken to you about the rest of this now. What you can see is that efferent arteriole folds back downwards, and it forms a network of capillaries that are tightly associated with the rest of the renal tubule, especially the proximal and the distal convoluted tubule, but also to a certain extent around the loop of Henle, although this doesn't show you that clearly here. And then all those capillaries join together and form a tiny venule, which joins other venules and eventually goes and forms the renal vein, which leaves the kidney. So I'm going to say that again. Okay, I think that by now you should be happy with the afferent arteriole. Afferent arteriole, capillary network of glomerulus, efferent arteriole. What I hadn't said before is that the efferent arteriole goes and divides into a network of capillaries which completely surround the tubule, especially the proximal and the distal convoluted tubule, before then rejoining and forming a little venule, which eventually will form the renal vein. This network of capillaries around the tubule is called the peritubular capillary network. Peri means around, tubular means tubular, capillary network. I hope that makes sense. Okay. All right. Look what I've done here. Repeated this twice. Who knows? Doesn't matter how many times I read through these things once I've dealt with them. They make sense, and I don't even notice until I'm busy telling you these things. Okay. All right. You do not have to know this name and this name. Okay. Just know that it's a branch of the renal artery and a branch of the renal vein. Okay. So here you've got renal artery going into the kidney, divides into many small, which divide into smaller, which divide into smaller, etc., etc. And this just shows you little afferent arterioles going into the glomerulus, but the glomerulus has been, the nephrons have been taken away except for this one here. It's just showing you the afferent arteriole going into the glomerulus. Okay, all right. Okay, so this particular one shows a longitudinal section through a renal corpuscle or a Malpighian body, and it shows you the podocytes. And don't obsess about these podocytes. Just know that they are funny little starfish-shaped cells. And in fact, and I'm going to say this at this point, but you don't have to know this, they don't lie flat. They actually each curve up and form and actually surround the capillary. That's not important. What is important though is the slits between the podocytes. You can see them here and here. Okay. All right. So this is a beautiful slide of a cross-section of the kidney cortex under a light microscope. How do you know that it's cortex? Because you can see the Malpighian body. So this is the glomerulus. Okay. All the capillaries of the glomerulus. This then would be Bowman's capsule, And you can see the space between the two layers of the walls of the Bowman's capsule. Okay, don't worry about that, don't worry about that. Then what you can see is there's a distal convoluted tubule in cross-section. There's a distal convoluted tubule in cross-section. There's a proximal, there's a proximal. So in other words, what this is if you took, let me go to the nice one, my favorite one. Okay, so if you took a section through here, okay, cut a line through there, what you can see is you can see the glomerulus, you can see the Bowman's capsule with a space between it. You can see a cross section through a proximal convoluted tubule, and then another cross section, and then through the distal. So I'm going back to that to show you that beautiful slide again. Okay, glomerulus, Bowman's capsule, space of Bowman's capsule. There's some proximal convoluted tubule, and I'll tell you how to tell the difference between proximal and distal now. There's another proximal convoluted tubule, there's distal. But because the cortex is made up of so many thousands and thousands of nephrons, doesn't mean that this proximal convoluted tubule belongs to this one, to this nephron. Might belong to another one. And then between them, you've also got all these tiny little blood capillaries. There's a blood capillary. There's a blood capillary. There's a blood capillary. There's another one. And those are all the little blood capillaries of the peritubular capillary network. Now, how do you know this is the cortex and not the medulla? Because it contains a renal corpuscle, and it's got distal convoluted tubules and proximal convoluted tubules. That's how you know that it's the cortex and not the medulla. Does that make sense? I hope it does. So this one as well, beautiful. Teachers who think that things like this are beautiful. Look at this, an afferent arteriole leading in. Can you see it? There's the capillary network. There's an efferent arteriole leading out. Here's Bowman's capsule. Okay, and you can see it as a funnel-shaped structure, as opposed to the previous slide, which showed a section across this way. So if you've got a section across that way, you're not going to see it as having like the opening of the funnel, the afferent and the efferent arterioles. I hope you understand that. And there's the proximal convoluted tubule leading off there. Okay, and here are some little blood capillaries. There's a capillary, etc. And then this slide, this drawing, shows you on the right side, it's a drawing of this. So what you can see is you can see the afferent arteriole. There it is on the slide. You can see the capillary network. There, you can see them on the slide. See the efferent arteriole on the drawing, efferent arteriole on the slide. You can see the Bowman's capsule, that filter structure, the outer wall, the inner wall, and the space. Outer wall, inner wall, space. And then you can see the proximal convoluted tubule leading off there. Proximal convoluted tubule leading off there. Okay, hope that makes sense. This particular technique is just where they fill a dye into the blood vessels. And so you're not seeing the tissues. All you're seeing is the blood vessels, the dye in the blood vessels. Here you can see an afferent arteriole. There. The glomerulus. And here, efferent arteriole. It's going to drive me nuts. Okay, right, does that make sense? And what you can see very clearly here, because you're seeing the dye, is you can see that the ball of the afferent arteriole is much, 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 much bigger than the ball of the efferent arteriole. because you're only seeing the dye, which is showing you the lumen of those. Okay. Now this is a scanning electron micrograph of a cast of glomerulus. So what they have done here is they've injected into the blood vessels a substance which then hardens within the blood vessels, and then they dissolve all the tissues. So all you can see is the latex or whatever it is they fill the blood vessels with, and you can't see any of the cells. So this shows you a glomerulus with the many capillary loops. Okay. And then this is a branch of a renal artery or a renal vein. This would be a renal artery branch. This would be a renal vein branch. Okay, and you can see the many capillaries of the glomerulus. And then this is the efferent arteriole leaving here. The afferent arteriole isn't shown as beautifully as it could be because it doesn't show you so nicely that it's got a wider bore than the efferent. And then here's a magnification 300 times. So this is an electron micrograph, scanning electron micrograph. So I could ask you, using this, the magnification, I could ask you questions about how wide is the bore of the efferent arteriole at this point here. Okay, and then you would have to use, you remember the formula you learned in grade 10, where you learned that actual size is equal to image size divided by magnification. So you would measure the image, you've got the magnification, and therefore you can work out the actual size. Okay, make sure you know how to do those. All right, I hope that makes sense. And just going to do one last slide, then we'll leave it because there's no written work for you to do today. What we're going to talk about is, here is the walls of the different regions of the tubule. Now, just like Bowman's capsule is made of a single layer of squamous epithelium, but it's a double-walled thing, so it's 2 layers. The walls of the rest of the tubule are also made of a single layer of epithelium. Okay, so let's have a look. Proximal convoluted tubule, cuboidal epithelium with a brush border of microvilli, and many mitochondria in those cells. So this top micrograph shows you a cross-section of proximal convoluted tubule. Look, you can see, because this little color key here shows that it comes from the proximal convoluted tubule. Right, cuboidal epithelial cells with microvilli. Loop of Henle is squamous epithelium. But as the loop of Henle goes back up, it becomes increasingly more cuboidal. So over here it would be cuboidal. Okay, so this is here, this yellow part, see, key, squamous epithelium would be loop of Henle, this part of the loop of Henle. Then as the loop of Henle becomes increasingly up towards the distal convoluted tubule, the green part, the cells become less and less flat, more and more cuboidal, no microvilli though. Okay, cuboidal, no microvilli. And then the collecting duct is cuboidal epithelium. So I'm going to go through that again. That's all that this says here. It says cuboidal epithelium with microvilli. If you're looking on the drawing on the left side, cuboidal epithelium with microvilli and many mitochondria in the cells. That's going to be important. Squamous epithelium, squamous, cuboidal. No microvilli, not as many mitochondria. And then cuboidal epithelium, no microvilli, not as many mitochondria, just a normal number of mitochondria. Okay, I hope that makes sense, and we'll end the lesson today. Please learn this work. There's a lot of terminology, and there's a lot of stuff you need to know to be able to understand the work for the next 2 or 3 lessons. Okay, all right.